All right, the last thing that we're going to look at is metabolism that is not working correctly, and we're going to look at the disease diabetes. Diabetes has two main presentations that we think about, and so if we think about what happens normally, we eat, there's blood glucose levels go high. In response to the blood glucose levels, insulin is secreted into the bloodstream, and then the cells are active through the insulin receptor, which binds, dimerizes, and through a series of events causes lots of things to happen, one of which is upregulate the transporter to transport glucose into the cell. We can also increase glycogen storage. We can also increase cell proliferation and growth, but they're all in response to the insulin receptor. Normally, in the type 1 diabetes, you can eat and you have lots and lots of sugar in the bloodstream, but what is absent? No insulin at all. And so insulin is not being produced. So if there's insulin not being produced, it doesn't hit the receptor isn't hit and activated, then we don't upregulate the transporters to actually lower the glucose levels quickly. Type 2 is where we have lots of blood glucose and we have relatively high levels of insulin, and in some reason the insulin may even bind to the receptor, but it doesn't cause all the signaling that allows the transporters to transport glucose nor any of the other processes. Okay. Now, one is called juvenile, one is what's called adult onset. Why in the world will we call type 1 juvenile? Type 1 sometimes is linked genetically, so your pancreas just doesn't secrete the insulin. Other times, you can actually have a disease, an illness, that can affect those cells, and then you just don't produce them. It's usually caught early. Type 2 was actually once really called adult onset. Okay, when did we usually think about seeing this? What kind of patient? Usually obese patients, usually later in life. Okay, and in some way, it seemed that their health later in life and the type of diet they had tends to make them resistant to insulin. Okay, and so it wasn't the fact that they weren't producing it, it was the fact that there was some sort of blockage in the signaling. We're going to talk about one of the theories about why type 2 diabetics or that symptom comes about. Unfortunately, we are seeing now the onset of type 2 diabetes in much younger patients, even now where it once was really later in life, 30s and 40s. We're now seeing them in teenagers, which is a big shift in when this disease is presented. Okay. Insulin resistance, there are a couple of pathways. I'm going to show one. This is one proposed mechanism. All right. And so again, let's take a look at the normal. Lots of glucose, lots of insulin. Insulin binds. The insulin receptors dimerize. They autophosphorylate. If we go all the way back to the signal transduction chapter, it then creates a binding site that recruits IRS1. Do you remember what IRS is? I know, it's where you pay money to the government. Insulin receptor substrate, okay? And the insulin receptor substrate binds, and the insulin receptor then phosphorylates it, which makes it the active state, and then it goes through a series of different pathways, whether it's to increase the glucose intake, or is to increase glycogen upregulation, or it could also go down and upregulate transcription factors to start cell growth. And so from that one signaling molecule, it can bind to other pathways, other proteins to cause that. All right, so it needs to be phosphorylated. Well, we talked about it being phosphorylated. It needs to be phosphorylated in the correct residue on that to cause that cascade. All right, so in the case of someone who has a diet that may not be well balanced, all right, and they are resistant to insulin, one thing that makes this disease state really bad is you might have lots and lots of sugar, but inside the cells, they feel like they're starving because they don't get the nutrients that they need. And so if you're not getting glucose, then they start mobilizing fat. And so signals are sending out, you're eating, you have lots of glucose in your bloodstream, but you feel like you're starving, so you release fat. The fats are carried by the albumins. The albumins are placed into the cells, and they can be converted and upregulate diacylglycerol, which will interact with protein kinase C. We've seen diacylglycerol and protein kinase C in the phosphoinositol pathway. If I upregulate the protein kinase C, it will phosphorylate the IRS1. But here's the problem. It phosphorylates it at a different site. When the IRS1 becomes phosphorylated in a different site here at a serine, it then is unable to be recruited to the receptor, which is being signaled, which has a phosphorylation, which has the binding site, which wants to phosphorylate IRS1, but this cannot be recruited. Thus, this cannot be a signaling molecule for the next steps. And so what we've done is we've used this pathway to negate one of the steps in the signal transduction pathway. And so this is one of the proposed mechanisms of why, even though there's insulin there, the receptor is not being, it's not actually acting in the way it should. Okay. Now, symptoms of diabetes. Typically, you can see high glucose concentrations in the blood. Since it's not being absorbed by the cells, there is one cell tissue type that will filter it, and that's the kidneys. And so what you're doing is you're bringing in the sugar, it gets filtered out in the kidneys, you can then excrete it in the urine, and so you get no nutritional value in that particular process. But as glucose comes out, glucose is very hydrophilic, and so it brings in water, so you become dehydrated, and so you tend to be very thirsty. You either have, in the case of type 1, no insulin in the bloodstream, or in case type 2, you have very high insulin but not processed. There's high levels of glucagon. That's the cell saying that they don't have any energy, so that's going to upregulate fat being broken down. Glycolysis tends to be inhibited. Gluconeogenesis and glycogen breakdown is stimulated to protect the brain, although we do have glucose and high concentration already. Leads to a constant feeling of hunger because you have a hormonal signal that says you need more energy or more glucose particularly, and thirsty because you're dehydrated. There is, in the adult onset, there tends to be weight loss, a marked weight loss. And so what you've done is you've substituted all the things that you're eating, and now you're breaking down your fat stores, although you are signaling starvation. And so that's something they also, a sudden weight loss, even though most of us would enjoy having sudden weight loss, is an indicator that maybe not something's not going quite right. Diabetes have a couple of drugs that we can use. First and foremost, it's insulin itself. For type 1 diabetics, but insulin will not work for type 2 diabetes as well. Sometimes it will if we try to overwhelm the system. There are a couple other drugs, one of which is metformin, probably the most famous one that is prescribed, usually given to those that are in a pre-diabetic state. They're right on the edge, and I'll talk about how we tend to find that out. But what they will do is they will stimulate the feeling or the concentration inside the cell. It'll raise AMP levels, okay? And if you raise AMP levels, it is similar to the same physiology that happens when you start exercising. And so if you start exercising or you use this drug to make it simulated like you have started exercising, AMP levels go up. So what happens in that case? Well, you start to break down the fats. You start glucose uptake by a different pathway, and you start having more mitochondria being formed in the muscle cell. So one of your largest cell types in your body is the muscle, and this stimulates AMP, which stimulates all your muscle going to absorb sugar in a different pathway than just insulin would. In the liver, I'm going to stop gluconeogenesis, which you really don't need to do. Okay, you stop fatty acid synthesis and you stop cholesterol synthesis in the signaling of the AMP. In adipose tissue, you stop fatty acid synthesis and you stop breaking down triacylglycerol, so you stop sending out fats. And in the heart muscle, you will also uptake glucose, you'll uptake glycolysis, and you will start fatty acid oxidation. And so you tend to signal another pathway to bring that glucose concentration down in another way, signaling not by the fact that insulin is there. Yes?
So, instead of giving a patient metformin, could you just tell them to exercise a certain amount every day? Would that be enough?
It usually is not enough, but you're going to give them metformin and you're going to wanting them to do exercise on top of that. And so it's together. Unfortunately, with patients that are in the pre-diabetic state or heading towards type 2 diabetes, exercise is probably something that they avoid anyway. And so the medication helps, but getting them to exercise will help also. And so sometimes a patient will take a pill better than going to the gym.
So, like the AMPK, so you're using that on anchor as well.
Mm-hmm.
But since it also, I guess, upregulates glycolysis, so you're creating ATP, you get higher concentrations of ATP. How does that be affected by the
Yeah. What'll happen is that drug battles. Okay, so it raises AMP concentrations, but then you got glycolysis going to make ATP. This is one of those things where usually in a biological system, if you make something and break it down, it's this futile cycle, right? But in this case, what you're trying to do is get the glucose level concentration down. So making that futile cycle where you make AMP, then you try to remake it, and by bringing in the glucose, you get the disease state, the high levels of glucose, down to normal levels. And so you're kind of tricking the body into going into something that it shouldn't, but it creates a benefit in the end. Any other questions? All right. Now, how in the world do we know that we're in a pre-diabetic state or not? Okay, so because glucose is not absorbed in the tissue, you tend to have fats and amino acids in the bloodstream. Once the fats get there, you tend to get, in the liver being mobilized, the synthesis of acetyl-CoA, which is not necessary for the citric acid cycle. So what happens is we create ketone bodies. And if you create ketone bodies too fast, they can't be converted into the forms that are being sent out, like the hydroxybutyrate. And what happens is you build up a concentration of the ketone bodies in which it undergoes a non-enzymatic reaction, and you produce acetone. And so another indicator of either someone who doesn't know they're diabetic, or if they are diabetic and non-compliant, you can actually, and they talk about it, a sweet smell. Okay, most people haven't been in an organic lab and haven't smelled the smell of acetone, but you can actually smell it off of their breath or even excreted through their sweat. And so you may recognize that smell, but most people will term it as a sweet smell. And we know that at that point, the ketone bodies are imbalanced. This is where we actually found ketone bodies, and we thought that they were a disease state and only a disease state. Later, we found that they were true energy molecules. Along with the production of these ketone bodies, there are a lot of acids bodies. And so we tend to, when you overproduce these, make the blood system a little bit acidic. And so we have an issue in terms of the acidity of the blood, which then changes the biochemistry in those environments. Glucose itself and hemoglobin. All right, a diabetic person that has high concentrations of glucose in their blood, I mean extremely high, will tend to allow glucose, and this happens to all of us. The glucose can be found in a equilibrium between the straight chain and the cyclic chain. And in the straight chain, it can actually react with the protein of hemoglobin, the N-terminus of the beta chain. They first form a Schiff's base, then it can react to create a very stable structure. Rearrangement can occur. All right. We measure this in the bloodstream, and it's called the A1C levels. Okay. And so we can measure how much sugar is attached to the hemoglobin. Normally, it should be in the 3 to 5% range. So about 3 to 5% of all of your hemoglobin should have, on the beta chain, a sugar attached to it. Once you get up to 6 to 15%, that's where we see the attachment for diabetics. In that range between 5 and 6, you're in the pre-diabetic state, and so this tends to creep up over time. Now, what has to happen for this to occur? The concentration of the blood glucose has to be high for a very long time, much longer than what you would see normally when you eat. And so it's an indicator that the blood glucose level has not been great. And so here's the deal. If you're treating someone who has diabetes, kind of like heading to the dentist, what do you do before you go to the dentist? Brush your teeth. You may even brush it, like a couple days ahead. I know I'm going to the doctor, to the dentist. They're going to clean my teeth. I want to be a good boy and girl when I go to the dentist. I might even floss once between the time, right? And then you get up there, oh, your teeth are so clean, right? Right. Well, here is the deal. What do you think you're going to do if you're a diabetic and you're heading to the doctor? What are you going to do in terms of monitoring your bloodstream and monitoring how much insulin you're going to take? You're probably going to be right on the button when you go in for your doctor's visit, right? Okay. Not always. Now, some people don't do that, but some people do. And so if you go in and you get a perfect 98 on the blood glucose, 100 is the stickler, all right? But you see 15% A1C, okay? What does that mean? They got it down good for the checkup, but for long extended periods of time, their blood glucose has been very, very high. Okay, what happens if they walk in and let's say they were 140 on your glucose level, but their A1C is down at 6? They might have just eaten before they got their blood glucose taken. But they've been very compliant because the long-term indicator was much lower than the short-term indicator. And so it's another way to monitor. Yes?
Reverse effects of type 2?
Can you reverse the effects of type 2? Yes. Yes, over time. And if you think about it, if it's going through that state in which you have lots of mobilized fats, a lot of people who undergo weight loss treatment, either surgically or under programs where they lose a lot of weight, they can tend to become much more insulin sensitive, along with changing their diet and changing their physiology in terms of losing weight. And so it is not absolutely irreversible. Type 2 can be. Unfortunately, it comes from a lifestyle habit. That particular disease comes more from a lifestyle habit than it is from a genetic predisposition for that type of thing. All right, here is a good indicator of what is different between the diabetic states. And so this is something that is a test that is taught in which to test how people are challenged. You can drink a glucose solution. It's this nasty, sickly, orangish drink usually they use, or a Coke, all right? And so you drink the Coke, and you can see that the concentrations of the sugar goes up very, very high, but over very short periods of time in a normal physiology, even though it spikes high, it gets down to what would be considered normal within 2 hours. Even go below and then come back up. But if you take a look at both type 1 and type 2 diabetics, you can see that the sugar goes high and stays high and stays high. Why would the sugar go down in a diabetic? Where's the sugar going down out of their blood? Where? In the urine. Okay, so it takes a long time for that to go. Where does it go in a normal person? All sorts of cells, right? Being used to be stored and or used at that moment in time. All right, if you take a look at both type 1 and type 2 diabetics, upon the injection of insulin, what do you see? Type 1 goes down, and type 1 looks exactly like what? Normal. That is a true indicator that the insulin was just not produced. Everything else was working, it's just the insulin wasn't produced. What do you see in type 2? Not a whole lot. There's a little, but not much difference. Depending upon how far you are in the type 2 diabetic place, a little bit of insulin can tend to give a little bit of relief, but you can get to a place where you're totally insulin independent and you can't control it with that. And so we use some of the other drugs to try to manage that.